Good afternoon, everyone. Myself, Dr. Nannu Shafaqatullah, here to present a research paper on the topic entitled A Novel Lactobacillus gasseri MF1 Strain Mechanism of Probiotic Action in the Prevention of Colorectal Cancer. Probiotics, these are live bacteria which when we administer or when we intake along with our diet, they're responsible to give a health benefit on the individual. These lacto-organisms include the strains of Lactobacillus, Lactococcus, Bifidobacteria, Streptococcus, and so on. They can survive easily in the intestine, they can proliferate and colonize in the intestine. They have no pathogenic, no immune reaction, no toxic, no allergic or mutagenic effect on the individual. These organisms are genetically stable, no plasmid transfer from these organisms to the pathogenic organisms which are present in the intestine. They have the defense mechanism in the intestine where they can stabilize the local microflora. They have the effect on immune modulation. They are responsible for the production of varieties of secondary metabolites which are responsible to give a health benefit on the individual. The most important effect of this probiotic is anti colorectal cancer effect. So colorectal cancer, it is the third most common cancer in the incidence as well as the mortality rate, and uh, around 8 percentage of the death due to cancer, it is because of the colorectal cancer. The treatment for colorectal cancer which is available, we can say that we have the chemotherapy as well as radiation. But this chemotherapy and radiation treatment is responsible to cause the side effect on the individual. So there is need to go for alternative therapy to treat this colorectal cancer. So it has been found that by many researchers that the endogenous microorganisms are responsible to cause they are responsible to cause the cancer. So this makes it reasonable to think that changing the intestinal microflora could influence the tumor development. So in the present study, we have isolated a organism Lactobacillus gasseri, and this organism has been used to find out its effect on the colorectal cancer induced mice. The bacteria or the probiotic bacteria has been isolated from the marine fish. It has been identified based on the morphological, physiological, and biochemical characteristics. Then the isolated organisms were subjected for probiotic properties. So around 14 different probiotic properties have been studied, which are based on the guidelines given by ICMR as well as FEO. So from this we could select only one organism as a potential probiotic organism, and that organism has been subjected for 16 rRNA sequencing, and finally it has been identified as the Lactobacillus gasseri strain MF1, and the sequences have been deposited in the NCBI GenBank database. The experiments on the mice were conducted based on the guidelines of Animal Ethical Committee. The animals were housed in the departmental animal house, and required conditions have been supplied to the animals. We have done the test on 25 organisms, 25 mice, and they were grouped into 5 groups. Each group comprised 5 mice, and the experiment has been conducted for 28 weeks. The animals were divided into Group 1, Group 2, Group 3, Group 4, and Group 5. Group 1 animals were the negative control, Group 2 were the positive control, Group 3 were the pre-treatment group, then Group 4 we have the post-treatment, Group 5 we have pre as well as post-treatment group. So the pre-treatment group is where the Lactobacillus gasseri has been given to the mice first, then it has been induced by DMH. In the post-treatment, first the organism have been induced by dimethyl hydrogen, then the Lactobacillus gasseri has been given, and in the pre and post treatment groups, so the Lactobacillus gasseri has been given first, then induced by DMH, then again the Lactobacillus, gasseri has been given to the animals. So the dose and drug and dosage preparation was according to the standard protocol, and subcutaneous injection of DMH has been given around 20 mg per kg body weight. Subcutaneous injection has been given, and the probiotic concentration of around 2 into 10 raised to 8 colony forming unit has been given, suspended in the milk by Galvez twice a week throughout the experimental period. The following parameters have been studied, that is the general health of the mice, food intake, body growth, as well as a survival study has been done. Tumor assessment has been done, fecal microbial count, antioxidant activity has been studied, as well as the bacterial enzyme activity has been studied. So in this study we have done only the preliminary studies. We have not gone for any advanced test. So in the food intake as well as the body growth and the survival study, we have seen from the graph, you can see that the pre and post treatment group have shown the maximum growth compared to the negative control, whereas the positive control and the pre and post treatment group has shown less in their body weight. The tumor assessment have been done. So in the positive control, we have seen around 10, that is the average 10 tumors have been seen in the positive control group, whereas in the pre and post treatment group, less than two, average two tumors have been found. The fecal microbial count has been studied. The pre and post treatment group, you have seen an increase in the lactobacillus organism, whereas in the pre and post treatment, the coliform number of coliform have reduced. Antioxidant study has been done. In this antioxidant study, we have studied about glutathione S transferase. So in this organism, in the pre and pre-post treatment group, we have seen increase in the concentration of increase in the effect of glutathione S transferase has been identified. The second one was superoxide dismutase assay. Here also in the superoxide dismutase assay, the increase in the function of superoxide dismutase has been identified in the pre as well as pre and post treatment groups. The third one was reduced glutathione assay. Here also the reduced glutathione activity was increased in the organisms, that is in the pre as well as pre and post treatment groups. Then we studied about the bacterial enzyme activity. In the bacterial enzyme activity, we have studied about the beta glucosidase activity. And the beta glucosidase activity was reduced in the pre and pre-post treatment group. Next was the beta glucuronidase activity. So here the beta glucuronidase activity was also reduced in the pre as well as pre and post treatment groups. So in the conclusion, we can say that the probiotic confer anti-carcinogenic effect by excluding the pathogenic microorganism by competing for nutrients and receptors, as well as they are responsible for the production of antimicrobial metabolites. In turn, they are responsible to activate the immune system. So here, whatever study we have done, it was just about the pre-study. So for the successful translation of these preclinical strategies into clinical practice will depend upon the outcomes of the clinical trials. So in the recommendation, there has been less work done on the probiotics from the non-fermented foods, and there is less products are available in the, that is the less probiotic products are available in the market. So these are my references, and thank you.